Hello, welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at a core practical in topic 6, which is using gel electrophoresis to separate DNA fragments of different length. This is often a practical that a lot of people struggle with, so it is quite good for you to review this. The questions for this practical are very, very repetitive and have been seen so many times in past paper questions. So once again, making it more important. So why do we do this? So DNA profiling is carried out to make millions of copies of a specific DNA sample through the process of PCR and gel electrophoresis. And this is usually used mostly in crime investigations and also to determine genetic relationships between organisms. So what do you need for this practical? You will need restriction endonuclease enzymes, agar gel, a gel tank, electrical supply, micropipettes, DNA sample, loading dye, UV light camera, buffer solution, DNA restriction ladder, and a beaker. So let's go through the setup. So the DNA sample is mixed with restriction endonuclease enzymes and a loading dye in a beaker. And the reason we add dye is so that we are actually able to see what's happening. The agar gel is prepared, and this is usually done by the lab tech and is poured onto the electrophoresis mold. The electrophoresis tank is filled with a buffer solution, and we use micropipettes to load the restriction ladder into the first well. We can then follow this up by loading the DNA samples you want to investigate into the other wells. So just similarly to what you did with the restriction endonuclease enzymes, you're going to do the same with the other DNA samples. And restriction enzymes are used to cut these DNA samples at different lengths. The tank is now connected to an electrical supply. So switch the supply on and leave until the dye has moved to the opposite end of the gel tank. The power supply is now switched off and disconnected from the electrical supply, and the gel from the tank is carefully removed and viewed under UV light for further analysis, depending on whatever your overall aim is for the practical. So what will you observe? So it will be observed that the lighter the DNA strand is, the further up it will travel to the opposite end. The DNA restriction ladder, which is what we loaded first in the first well, can be used to measure the size and also observe similarities of the different fragments. DNA is unique, and this is because there is a wide variety of introns, which is the coding parts of DNA, and STRs, which are the short tandem repeats. The restriction endonucleases cut the DNA into fragments, and the phosphate in DNA nucleotides carry a negative charge, giving it an overall negative charge, like the overall charge of the DNA. And this is what attracts it to the opposite end of the tank, and that's because there's a positive electrode at the opposite end, and as we know from GCSE, opposite charges attract. And this is why this happens when the power supply is switched on. So varying STRs at different loci lead to different distances covered by the DNA segments, resulting in a unique DNA profile being produced. I'm Professor SNAB today. Hope you found this video useful. If you have any questions, please let me know in the comments below and I'll get back to them as soon as possible. And don't forget to like, share and subscribe.